I'm going to talk about genome structural variation and phenotypic diversity in soybean. So I started working on soybean in 2008, and I came from a maize and potato background, but mainly working on gene expression. And I've moved more into the genomics realm of just studying DNA and sequence variation there. So it's a little bit different type of work, but I think there's a lot of great opportunities to do this type of work in soybean for lots of reasons, and I'll try to touch on that all today. So this one didn't move. Is this one supposed to move too?
It's sluggish.
Oh, there we go. Okay.
Attention to it.
Okay. I'll just look at that one. Okay. So soybean genome, some of you may know, in 2010 the sequence was published in Nature. So in some ways it's a relatively new genome still. It's been around a couple of years, and it's about a gigabase, maybe 1.1 billion, and so it's a very good system. If you're going to work on legumes, it's a very good and very complete genome compared to other opportunities that are out there. But as a genomicist working on a genome that's sequenced, what am I supposed to do, right? We've answered a lot of questions already in this community. So the questions that I'm really mostly interested in are touching on more areas of genomic variation. And we have a reference, but how much does that tell us about what's actually in the germplasm? And moving beyond that, beyond what's in the existing germplasm, how can we maybe target new variation for trait improvement? So I'm sort of involved in studying natural variation and induced variation as well, and also some targeted stuff that I'm going to talk a bit about at the end that involves more of the genome engineering aspects. Okay. So a lot of times when we think about genetic variation in plant species, the majority of the work goes into understanding variation based on SNP and indels and those types of studies. And of course, because in plants we have a deep history of working in transposable elements, that's also an area that gets quite a bit of attention, even in the soybean community. There's people who specialize in this area. When I started out a few years back, it didn't seem like anyone was really doing much work in this area. Structural changes, meaning large deletions, duplications, inversions, and translocations. I have a cytogenetics background. Perhaps that's why I was attracted to this type of research, because this is sort of what cytogenetics is becoming, sort of a high-resolution way to detect structural changes in genomes. So just for a little background, what do we know about the soybean genome and structural variation at the genome-wide level? Well, the earliest study that really tried to do a very good genome-wide assessment of structural variation in soybean was done by a Korean group, Soo-Ka Lee's group, this Kim et al. Paper, published in 2010, where they looked at, basically they just did a lot of resequencing of a wild relative called glycine soja, or soya, depending on how you pronounce it. And they found a lot of structural variation. They actually focused most of their attention on structural variation. So they didn't do a de novo assembly of this wild genome. What they did is they basically compared it to the reference of soybean, and they found things that they focused a lot on what was missing in the soya genome compared to glycine max, which is soybean. So here you see an example of sort of the structural changes that they defined. So first they, of course, did a lot of nucleotide changes, things that you might expect, SNPs that affect protein structure, are predicted to affect protein structure, frame shift indels that may be sources of load, of genetic load. But then they spent a lot of time talking about the events that were found, genes that were found in glycine max that were completely missing in glycine soya. So in this case, they found over 700 of those in these two accessions. And then they found some evidence of genes that are in glycine soja that were missing in max entirely. So this is what we would call presence-absence variance. Okay? Sometimes people use the term copy number variance. Presence-absence variance is sort of a subset of that. It's cases where you have genes that are completely missing and present and absent from one line to the next. Okay? So I focus my research quite a bit on that. And then there's inversions that they defined, and they also defined or talked about potential translocations as well. So those are all different forms of structural variance, basically what you see here on the right side of this diagram. Okay, so does this stuff matter, is the first question we might want to ask. Why even research it? Well, there's a lot of evidence that it does matter. If I'm sure there's people involved in these projects that are sitting in this room. The GWAS studies in maize have found that there's an overrepresentation of structural variants are associated with phenotypic changes at a rate that's much higher than they would expect at random. It's like 10 or 20-fold greater than they would have expected. So it appears that these are very phenotypically important events that occur, at least in the maize genome. And so this is obviously an area that we want to explore as well in the soybean genome. So what work has been done beyond this paper in 2010? I'm basically going to focus on. This is sort of my introduction. This is the work that I'm going to show you today. I'm going to talk a bit about some of the work we've done to develop platforms where we can study mutagenesis, induced variation, structural variation in the soybean genome. So we're going to focus on fast neutron, mutant lines, and basically under the paradigm, can we associate phenotypic changes with some of the deletions that are caused by fast neutrons, and also the general questions of genome stability in soybean using this population. We're going to talk a little bit about some of the early microarray work. Well, actually, I'm going to skip this part, but I should mention that we did work on structural variation within accessions. So within the reference genotype, Williams 82, we had a paper that came out a little over a year ago on that topic. And then I'm going to focus most of my time on the focusing on comparing 4 soybean genomes, exomes in this case, where we found a lot of gene content variants. So we started to look genome structure-wide or genome-wide for structural changes in these genotypes as well. So these are the sort of the topics that I'm going to cover today. So this is sort of the natural variation piece. This is the induced variation piece. And at the end, I'll talk a little bit about genome engineering, targeted changes. Okay, so let's talk a little bit about this fast neutron business. So the soybean community has what some would consider very weak genetic resources to do functional genomics. So there's been an investment in recent years in developing mutant lines that are going to be useful for this purpose. And so my group has been involved in helping to develop genotyping platforms for fast neutron mutations. So fast neutrons, as many of you know maybe, is a forward genetic approach where you can expose your seed to fast neutron irradiation, and then you come out with lots of new phenotypes. In fact, your most common phenotype is dead because it does so many bad things to the genome. But we also recover many things that look quite interesting, like in this case, you see somewhat of a mosaic chlorotic issue. You have a dwarf chlorotic issue here. You have what appears to be a short petiole architecture mutant in this picture here. And here you see a mutant that has reduced trichomes. You can see wild type here versus the mutant. So these are the sorts of things we see in the field. And of course, we also assess these things for seed composition changes. But the reason really people care about this type of work is because maybe we can find protein and oil mutants that have elevated levels that might be useful for breeding purposes. Okay, but these are the easy ones. These make nice pictures, so I show you these. So we developed a platform commonly known as comparative genomic hybridization, or CGH, that sort of works this way. It's a microarray. So here you have our parent line. This is the wild type, and this is a fast neutron line that we've irradiated. What basically happens is a fast neutron comes in and, this example, blows a single deletion in chromosome 11. Okay? So then we run a microarray of these two versus one another. Oh, sorry, I forgot to say, this is forward genetics. So first we have to find a phenotype that we care about. So here you see a late maturity mutant here in the field. You see everything else is nice and brown in the background. This plant doesn't look like it's developing quite at the same speed. And so what we do is we can label, it's a 2-die system, so we can create a microarray that basically tiles the entire genome. So we have a probe that goes every about kb along each of these chromosome arms. So there's chromosome 1 and 2 and 3 and so on. We could pretty much tile the entire array this way, or the entire genome this way. And so each of these spots represents a 1 kb interval. And we label our control with Psi5 and our, yeah, that's right, and our fast neutron line with Psi3, and then we hybridize, and we look for spots that basically tell us where the deletions have occurred in our fast neutron line. And then we can represent them graphically. It kind of makes a nice picture. So here for this example, I've just made fake data, where the log ratio between the two lines is essentially 0 for everything, and then we have a nice, clean 10 kb deletion shown here on chromosome 11. Okay, does that make sense? Good, because I'm going to show more pictures like this. Okay, so that's where we might detect deletion. We can find a whole range of things. We can find small deletions where very few probes are affected, and we can find sort of a 10 kb type thing, or we can find sometimes half a chromosome maybe has shown some sort of deletion. Like I say, these fast neutrons, they create all kinds of problems in the genome. We can find I'm not showing it, but we also find duplications sometimes, all kinds of craziness. And then we find most commonly in most of the lines that we look at, we have more than one locus that has some sort of change. So that's kind of a complicated genetic system to work with. Okay, but it's really quite nice in some ways as well, because every now and then you get a phenotype that you really, really care about. This is an X-ray line. It's not a fast neutron line, but it's an X-ray irradiation line that was developed a few years ago that has, we use CGH here. This is in collaboration with Gary Stacy's group, where they have a high oleic acid mutant, which is an important trait in soybean. And they did CGH on this particular line up in our lab and found a very specific deletion that occurred towards the end of chromosome 10. And so it turns out that this is a line that they could then create primers for to flank that deletion breakpoint, and they can amplify that, they can detect where the deletion exists in those lines. And then they'll sequence the PCR product. They'll know exactly where the deletion occurred, the breakpoints, and they can also then use this as a marker for, if they want to use this to integress the trait. Because as you can imagine, you don't want to have to run seed assays on all your backcross population, so you can just use this marker now. So it's a nice system if you have this sort of trait that can be easily scored with a PCR assay. So anyway, that's just an example of how this is useful. And this is all published in a paper last year, if you want to read more about it. So this turns out to be a 163 kb deletion, which is actually relatively modest in terms of some of the deletions we see. But there's more than, there's probably about, 5 or 6, 8 genes in that region. Okay. So what types of variation are we generating using this deletion mutagenesis in the fast neutron population? Well, here's a kind of a neat example. This is plant architecture mutant. We've done hundreds of these lines. We've actually done a couple hundred on the CGH platform now. I'm just going to show you one nice example. So this is an example of an architecture mutant that has very narrow profile. It has a very short petiole. So we're interested in using it as a high density planting line. And so what you see here is basically, you can see how sort of short these arms are. In early development, they can be planted quite closely to each other without having any sort of detrimental effect on one another. So we call this the Christmas tree mutant, because it looks kind of like a Christmas tree when it's early in development. So we did CGH, and this is an old picture. It's not the prettiest one we've ever done, but it's a nice example of what we could find. So it turns out that you can see a really conspicuous deletion here on chromosome 13. So we thought, oh, this is simple. We just found the causative mutation that causes this phenotype. But it actually turns out that that wasn't the mutation that caused the phenotype. We did a little follow-up work on it and found that there's also a small deletion here on 17 that actually segregated perfectly with that phenotype in a Mendelian fashion. Anyone want to guess why it's so small? Or at least the profile is so small? Turns out it's a dominant mutation. This is a heterozygous plant that we scored. So there's a little bit of work that went involved in following this up. But again, we were able to create primers. We detected the breakpoint, and we could look at a segregating row, for instance, and quickly determine the causative mutation. So now this has become a plant that one of my students is interested in trying to breed with. So he's starting to cross that chromosome 17 locus into some different backgrounds to see if he can create a narrow spacing soybean plant. Okay. So anyway, in summary, the fast neutron resource is sort of, I show these slides in part because it was a takeoff point for studying natural variation. But to date, this has been something that we're still continuing to work on. We have over 20,000 fast neutron lines that have been phenotyped for seed composition morphology. So that's just a lot of row walking forward genetics. And actually, within the next month, we'll be up to 200 lines that have been genotyped by CGH. We're starting to get some deletion associations and trait associations for these lines, and we're starting to compile all this data, and we're going to try to write up a paper soon on the genome resiliency towards fast neutron irradiation, the types of, and the locations and types of changes we see, both in terms of deletions and duplications. Okay, so most of you are probably thinking, great, but I'd never want to work with a fast neutron line, especially if you're interested in functional genomics, because it's a sloppy background. I just admitted that. I mean, our Christmas tree mutant, that deletion, it's a dominant mutation. It's about 700 kb though. There's, I don't know, 50 genes or something in that space. So maybe it's not a perfect system for forward genetics, but it was a great system for developing platforms to study structural variation. So what about natural variation? That's what most people really care about, so I'm going to talk about that next. Okay, so when we think about soybean genetic diversity, there's been sort of this, the dogma that's built up over the years is what I'm going to very briefly present. And Jeff, you can correct me if I misspeak on any of this. You've been in the community longer than I have. So we know that there's a lot of phenotypes, quite a range of phenotypes in cultivars. There's maturity differences, there's from the landrace through the modern elites, there's quite a bit of changes that have occurred. However, from the molecular perspective, those who've studied this, basically they say it's a pretty bottlenecked germplasm. Our SNP rate is very low, compared to something like maize. We get a SNP of maybe 1 per kb, something like that. So people have built up SNP platforms to study this, and this is a really big initiative in the community. They're taking pretty much the whole GRIN germplasm. So we're talking about 20,000 lines. They're going to put them all on an Infinium chip, and they're going to genotype, and they're going to develop these sorts of profiles that we show here. So we did this for a very small number of lines. This is a 50K chip where we could detect basically what you see in black is SNPs that are polymorphic to the reference genome. So the reference genome is Williams 82. And so this is Archer versus Williams 82. And then matches are gray and polymorphic are black. And so you can see some chromosomes are very similar. And this isn't really what we're interested in, but this is what the community is building. They're building a whole database on just this sort of haplotyping of all these accessions. But what we've been doing is we've been asking the question, is structural variation also low? And if so, that's okay, but where does it persist? And if not, is it something different than what we might have expected? When we first started doing these experiments, I should say, people who were sort of higher up in the community basically said, I'm not sure that's worth your time. You're just going to see that everything's the same. You're not going to have any structural variation in these genomes. It's way too bottlenecked. But it turns out that we started doing some sequencing work on exomes. So we did exome capture and started sequencing some of these genotypes. So again, this is our core group that we work with, Williams 82, Archer, Noir 1, and Minsoy. We found when we did this that there was what we call presence-absence variation in this very small sample. So here you can see an example here of Minsoy that has 79 genes that are presence-absence variants compared to these other 3 lines. So 36 of these are present only in Minsoy and are missing in the other 3, and so on. So you can interpret this as sort of messy. But basically, the long story is, made short, is that 133 genes had some sort of presence-absence variance. And this is using very high threshold statistics. So the real number is probably something like 500 or, using the sort of the canonical 46,000 genes that have been described. I'd say about 500 of those are probably in this category, but we were only able to detect ones that were very unique. And so I won't get into there's a lot of duplications in soybeans. There's a lot of genes that weren't part of this analysis. The quick story is, though, that we're detecting a lot more than we expected, okay? So we did some PCR confirmation of this, and you can see, these are just some examples. Each glyma represents a different gene that's been described in the Williams 82 reference. And you can see our core group is here, these first 4. We're scoring presence-absence with PCR, and then we have a set of, I think, 27 additional lines that we scored just to get a feel for how common and rare these types of variants are. And you can see we see a wide range of how common these are. So in this case, Archer, on this particular glyma on chromosome 5, is the only absent line. The rest of them are all have present. So this might be a rare variant, a rare structural variant that's missing just in a few lines. Whereas in others, you could see that there's like a 50-50 type of presence-absence frequency. So this is, again, small sampling, small number of genotypes. But we only used 4 to find this core group. So you can imagine if we had done a bigger set of germplasm with the exome, we would probably find a much more extensive set of genes that have this type of variation. Okay, so we decided to go genome-wide with this and started using our CGH platform because we can only do a few exomes this way, a subset of genes, but we wanted to do intergenic space and we wanted to do all the genes. So we started to apply our big chip to this. And it turned out that we could actually make out features quite nicely using this approach. So here you see an example. This graphic depicts chromosome 3, a comparison of Noir 1 to Williams 82. And so basically what you see here is the top of the chromosome, so that's position 0 up to about 48 megabases. The quick interpretation, I don't want to get too nuanced with this because it's hybridization based. I know there's going to be some fuzziness in the data, but the quick interpretation that I think we're seeing here is that down clusters, basically things that fall off this line, are where Noir1 has a lower signal than Williams-82. Most of those cases, many of those cases, Noir1 will not have these sequences. Those are going to be presence-absence variants, absent in Noir1, present in Williams-82. Okay? And then the rare up clusters we see, like here, these are probably instances where Williams-82 does not have these sequences and they are present in Noir1. Now, we don't see that very often. Why do you think we don't see that very often? There's a really good reason.
Isn't Williams-82 the reference genome?
Bingo. Exactly. So how would we see that at all? That's a great question. We were really shocked when we first started seeing this. Well, it turns out there's some issues with the assembly in certain regions of the genome. So you can read Hahn et al. 2011 if you're really interested in learning more. But basically, yeah, there's some issues where Williams-82 has, there's heterogeneity between certain lines of Williams-82. So any given line of Williams-82 that you're hybridized will either have or not have certain features in this front part of chromosome 3, the top half of chromosome 3. So it's complicated. It's also somewhat possible that some of these up C and V are true copy number variants where they're present in Williams-82, but they just have more copies in these other lines. We think we're probably not detecting too many of those. Our chemistry isn't really very good at detecting that sort of thing, but it's very good at detecting presence-absence variants. Okay, so this is just a really quick snapshot of 20 chromosomes by 3 comparisons. They're all referenced to Williams-82. You've got Archer, Minsoy, Noir1. And you can see, I'm not going to get into the details here, but basically some chromosomes have a lot of variation. Some of them have very little, so on. Okay. And again, it depends on the comparison you're making. However, there are even on this small sampling, we wanted to pick out some of the general trends that that are sort of obvious even to the eye. And so we can sort of detect where hotspots may exist. So here's just chromosome 3 showing Archer, Minsoy, and Noir1 all compared to Williams-82. And then I just made a quick composite of that. So I just take all 3 of the variants of the data points over a sliding scale, and you can sort of create this easy depiction, visual depiction of where variation exists between the 3 genotypes, all compared to Williams-82. So here you see in the front half of this chromosome, top half of this chromosome, you're picking up some of the variation here that you can see by eye with the peaks. And the same is somewhat true on chromosome 18. It's another example where you get a nice difference between those 3 genotypes, and then the rest of the chromosomes relatively flat except at this end. Okay, why am I doing this? It's because I want to reduce those 60 windows down to 20 so we can digest this a little bit easier. Okay, so this is a quick aside that I think is somewhat interesting. Maybe some of you who followed this literature know that there's been a lot of work in structural variation done in maize. This is what maize looks like. You notice the difference? This is a similar set of germplasm that they're comparing here to their reference, which is B73. Basically, in maize, here you got a little pocket of conservation, but everything else is just crazy structural variation. Really, anyone who's done work on structural variation, nobody's ever seen what they see in maize. The rates are just so much higher. Maize is a freak, let's face it. It is. I think a lot of us knew that anyway, but this is just further confirmation. So it turns out that there's some association with these regions of conservation, maybe domestication, loci. So this is sort of the opposite. We were talking to Jeff about this last night. It's sort of the opposite profile that we would see in soybean. In soybean, the rule is sort of very conserved structural variation with these nice dashing peaks. Here you see the opposite, lots of variation, and then some rare conservation. Yeah?
So are they really hotspots, or are they identical by descent?
You mean in soybean? Or?
Yeah. Are they old and common, or are they really truly hotspots and they're all unique within that?
I see. You're talking about the soybean peaks.
Yeah.
Well, I'd say we don't really know because our sampling's too small at this point. But I think we can probably answer that question within the next 6 months because we're going to do a great deal more sampling. So hold tight. Hopefully we'll get there. But anyway, as I said, it's kind of interesting. It's just sort of from a philosophical standpoint. You could ask very different questions of these 2 species depending on what you're really interested in. So anyway, I'll get rid of maize now, and we'll talk about soybean again. So in this case, we can sort of circle where hotspots occur, and I'll just call them copy number variation hotspots. It's sort of the accepted term. It's really not the best word, hotspots, but we'll just go with that for now. And so the next question becomes, well, if we look genome-wide, so I just showed you 2 chromosomes, let's do this for the whole genome. We can sort of, again, we can set cutoffs, or we can just sort of visually assess where these occur, and we can start to associate certain gene classes with these regions of structural variation. It turns out when we do this, when we look for PFAM families that are enriched for this sort of, in these sorts of regions, it turns out that there's very little that is associated with these structural variation regions. But when we do find the classes, it turns out that there's maybe some kind of obvious classes that jump out. So like the nucleotide binding domain family and the receptor-like protein families turn out to be very commonly enriched for these regions. I think it's like 10-fold or 15-fold. I can't remember the exact numbers, but it's very, very high compared to everything else that we've looked at. So these are, as you might suspect, these are things that are involved with our gene clusters. NBS, LRR domains, those sorts of things. So they're basically resistance gene loci. And so people who've studied soybean know that this is, the people who worked on this a long time, they know this is group N. So they linkage group N, and we know that the linkage group N carries a lot of resistance genes in the germplasm. So it turns out that that region also carries a lot of structural variation. Same can be said on chromosomes 16 and 18. I don't know what the letters are for those, but the people who do this stuff know that this is exactly where these things are found. Okay, so this is quite interesting. We have gene content variation. We can reveal many of these genes that are present in Williams 82 that are missing in cultivars. That's probably the easiest thing we can do because of the platform we're using is based on Williams 82. What we don't know is the other half. What's out there that isn't in the Williams 82 genome? I show this to you because it emphasizes how good we are at detecting what's not in the reference, but how poor we are at detecting the rest of it. And there's probably a lot of variation out there. There may be hundreds of different alleles out there that are essentially undetectable at this point because we're married to the reference. Okay. So, you can do de novo assemblies of these regions. How do you do that without breaking the bank? That's my phone, by the way. I'm not going to blame any of you. I guess I forgot to put it on vibrate. All right. And it's unknown, too. So these are the kinds of questions that we're very interested in doing. Very difficult to get to, though, as you may imagine. OK, so back to the heart of the question. So is there any evidence that this is real? What work is out there to tell us that copy number structural variation actually is associated with biotic defense? Well, it turns out that very timely, in fact, Andrew Bent's lab at Wisconsin very recently published this work on the soybean cyst nematode locus RHG1. So I recommend anyone who's interested in structural variation or just plant pathology in general looks at this paper because it's really unique. They found that in a susceptible Williams 82 line, they have about a 30 kb motif that, it's a region of about 4 or 5 genes that is present in a single copy in Williams 82, which is a susceptible line. If you look at Peking, which is somewhat resistant, they find 3 copies of that 30 kb thing in tandem. This is fiberfish data, by the way, in case you're wondering how they figured this out, this arrangement. And then in the highly resistant line, Fayette, they found 10 copies of this actual cluster. They're not NBS-LRR, but they're still copy number variants that they're associating with resistance. And when they looked a little more depth in this area, they found that they're making, at this point, somewhat loose associations that the number of copies are raising expression and raising resistance. So this is, we just wrote a paper recently about structural variation. There's very few people who've been able to do this sort of work, to actually associate copy number with a very important phenotype. This is like, in soybean cyst nematode, in the soybean world, that's like the Alzheimer's. That's a big deal. This is like the #1 biotic stress that people care about. So this is a very important work. Okay, so that's some nice direct evidence that structural variation does matter for biotic defense. We think this is going to become more and more common. If you talk to people who've done this type of work, people working on rust, people working on, well, there's a couple different pathogens that people are working on across the country. They're starting to sequence backs. between resistant accessions versus, and they're comparing to the Williams 82 type, they find this sort of structural variation. It's very difficult to sequence through these areas even with a back-by-back type method because structural variation is quite profound. Okay, so when we think about this, let's bring this back to perspective. So, conditional genes. These genes are quite conditional, right? It depends, how important they are depends a great deal on the environment that the plant is growing in. Whether or not they're important at all depends on the presence of a given biotic pathogen, be it soybean cyst nematode or a fungus, or oomycete, whatever. So it's really easy to think, well, why not just accumulate a ton of this stuff? If copy number is good and it goes up, and why aren't breeders just selecting for the most powerful biotic stress respondent lines and just pyramiding these genes? It's a legitimate question. I think part of the reason might be that there might be reason to select against these loci. There's evidence from the Arabidopsis literature that says that our genes can be associated with yield drag. This is, I think most breeders probably already know this, but this is actually direct evidence that an R gene per se has an effect on yield. I'll skip through the details on that, but if you want to, it's published a couple of years ago in Nature. Okay, so if breeders are sort of trained to select against, I mean, this is the theory, I suppose. Breeders are, whether it be through linkage drag or R gene activity per se, if these clusters do reduce something that breeders find desirable, like yield, is it a system that's sort of built to fail? If you're not selecting for the biotic stress response, are you going to then select against it? I guess that's the question. And I'd like to test that to some degree. So how can we test the effect of specific genes or clusters on yield? This is a really tough question. You could try to build isolines, I suppose, and you spend years back crossing, and you might get some answer out of that. And I think people have done that to some extent, and they've gotten reasonable answers. But there's new technologies out there that may allow us to ask these questions a little more directly. I'm going to talk a little bit about that now. Precision genomics. I only have a few minutes left though, don't I? Okay, I'll go really quickly through this. So the zinc finger nuclease work you may have be familiar with. It's basically it's a genome engineering platform that allows you to create double-stranded breaks at specific regions of the genome. And so here's an example. It shows a picture of this is a soybean gene. Say you want to mutate it here. You can create a double-stranded break that then, say, creates an indel. DNA repair is sometimes messy. It's usually good, but sometimes it's messy enough that you can create targeted mutations this way. So you basically engineer a protein that will specifically bind the sequence that you're interested in. That's the blue domains here. That's the zinc finger. Here's the nuclease in purple that creates the break. So engineers have gotten very good at this. They have informatic tools that tell them exactly how to design these blue domains here so that they'll bind exactly where they want it to in the genome. And so this is just an example of the vector. So this is what creates the blue domains. And then you can target those in soybean, and you can go in with your zinc finger. It's a transgene. You put it in the plant. And say you want to target this gene here. I'm just blowing it up. You can express that gene. You can go and create your double-stranded break on the other chromosome. That's shown here. And then you can create a point mutation. Okay, that's sort of the basic. That's the easiest thing to do with genome engineering. Okay, so this is nice because if you say wanted to mutate a gene that's important to you, it's going to be unlinked. The transgene is going to be unlinked to your mutation point. So you say you want to mutate FAD2. There's your gene that you care about. You can introduce the transgene. It goes into an unlinked region of the genome. It goes and creates its mutation, and then you can end up segregating out what you want. So now we've gotten rid of the transgene, which was heterozygous, and we've now made a homozygous mutant line. So this is sort of useful technology for this reason. We want to do, well, we've already done a bit of this, but we want to do a little bit more with it. So this is examples in soybean where we've created new alleles of dicer genes using zinc finger nuclease mutagenesis. And now we want to create constructs that will actually target two regions on linked, or two different locations on a linked region. So in this case, we just create twice as many nucleases, and we again put them in at a transgenic location, and then we can target potentially two regions of, say, an R gene cluster. I hope this makes sense. So we've run this transgene here. It locates this domain. We run this transgene here, it locates this domain. What does it do? It creates double-stranded breaks and potentially can create an isoline that doesn't have any linkage drag, basically. It can get rid of a cluster or can get rid of a gene. Or it could potentially, the other thing that's nice, I'm not going to show you this because it takes too long, but you can also create duplications of that cluster or inversions of that cluster. So you can create a whole, theoretically, you can create a whole slew of different types of structural variation events at those loci. Okay. And then again, they're isolines. You segregate out your transgene and it's going to be the line you started with minus your genes or plus your duplications, and you can actually bulk those up and potentially evaluate them for phenotypic changes. Okay, so at this point, this is just sort of some basics. We can create mutations in most of the ZFNs. We're starting to develop the reagents to create site-specific deletions as well. Eventually, the technologies will get stronger and better. These things advance. If you follow this literature of genome engineering, it's like every 3 months there's a new one-page paper in Nature Biotech or something that's advanced the technology, but it's incrementally getting stronger and stronger in terms of what you can do. So we're interested in targeting some of the R gene clusters for site-specific deletions, and then down the line we'll probably be able to do more sophisticated targeting as the technologies evolve. Okay, so conclusions. I know I've kind of rushed through the last few minutes of this, but I want to wrap up so that you guys can go back to class and whatever you need to do. So structural variation, gene content variation can be resolved at 3 levels in soybean that we're working on anyway. We're working on the random-induced deletion trait associations, natural variation, which to date has revealed a few things, but most prominently variation within defense gene clusters. And now we're starting to explore whether or not we can do targeted structural changes. Where and why is really the question. And what's our commitment to, you really have to care about what you're targeting because there's nothing easy about doing this sort of engineering. Anyone who's ever done a transformation experiment in soybean knows that alone is not trivial. But to actually then follow it up with the genetics and the evaluation of the plants for this sort of thing is a real commitment. Okay, so where are we going and what are we missing? I think to date, to assess structural, so the attractive thing to say is, well, microarrays are old. It's a dying technology. It's actually sort of true. Nimblegen just decided they're not going to continue our product line that we've been relying on for a few years. So maybe we need to go into genome resequencing to start to get a better resolution of what's going on. But even that's pretty limited in what it can do. Read length is, we're talking about 30 kb units that are repeated. There's no sequencing technology out there that's going to resolve that sort of thing. So we have to be creative about how we're going to be able to assess the types of variation that we're interested in looking at. And so I guess maybe to some extent, as powerful and as great as resequencing technology has gotten, it hasn't really helped people who are interested in this sort of question, because what it's done is it's increased volume of reads, but they've all gotten shorter. It's gotten cheap, but and so some people are developing algorithms to do sort of structural variation using sequencing technologies. But it's not an exact science either. I'm not sure it's that much better than microarrays at this point. So anyway, we'll see where the community goes in this direction. And I think we're going to be part of it, wherever it goes. Okay, quick acknowledgements. I know there's a lot of people. I'm going to highlight BJ Hahn for generating most of the data that you saw with CGH. Liam McHale at Ohio State, who helped with a lot of the analysis of the natural variation work. Carol Vance's group, who really developed the fast neutron populations. Lots of great collaborators up here I don't have time to expand on. Sean Curtin, who really has pioneered the precision genomics stuff along with Dan Voitas's group in translating their technologies to soybean. Oh, and funding sources, yes. A lot of it's come through commodity boards, but we've also had some help from the USDA and NSF to get these projects off the ground. So, okay, that's all I have. I'll be happy to answer any questions. Thanks.